Hello, everybody. Today I would like to talk about the management of the natural variability of plant-based pharmaceutical ingredients. And I will particularly make a focus on vegetable oils. But before starting, just a few words about us. ADM SIO is a manufacturer of specialty oils and fats with more than 60 years experience in their manufacturing and purification. We produce highly purified pharmaceutical oils for injectable, oral, and topical applications produced in a GMP environment. Our products are regulatory supported with type 2 and 4 drug master files and with certificates of suitability. So I will first talk about the reasons why it is of major importance to manage the variability of pharmaceutical ingredients. Then I will make an overview of the origins of this variability related to plant-based pharmaceutical ingredients with a focus on vegetable oils. And lastly, I will explain how we manage that variability from the raw material to the finished products to guarantee the consistency of our pharmaceutical ingredients. So why is it of major importance to manage this variability? You may know that pharmaceutical excipients, even if they have been for a long time presented as the inactive part of the drug, play a key role in drugs formulation. They allow the active parts to be delivered in a proper way. And their variations can impact the drug product performances, its bioavailability. They can also affect its physical and chemical stability. And they can also have an impact on the safety of the drug if they lead to the formation of potentially toxic degradants. And all these characteristics have a direct impact on the drug therapeutic effectiveness. And because of the important role they play in drug formulations, excipient quality is as important as that of the APIs. Over the last years, there has been an increasing focus on securing the quality of excipients, defining critical quality attributes and functional related characteristics. And this growing concern led to changes in the regulatory expectations. USP, for example, is more and more pushing towards higher quality standards and characterization of the excipients, adding to the product monographs a general chapter on excipient performance. This general chapter of the USP provides some recommendations for the characterization of excipients properties that are not evaluated in the monograph. The IPEC guidelines for GMP applied to pharmaceutical excipients has also recently been updated to take into account these new expectations. Now let's have a look at the origins of the variability related to plant-based products and in particular vegetable oils. So vegetable oils are natural products and by definition they are subject to a certain degree of variability. And this variability has many different origins. Agronomic conditions first. Variability can come from cultivation techniques like crop cultivars, sowing conditions, if the crops are irrigated, fertilized or not. It is also dependent on weather conditions and temperatures in line with the geographic area and the soil characteristics. And all these parameters play an important role on the oil content of the seeds and the fruits, but also on the fatty acid composition of the oils that will be extracted from these raw materials. After harvest, storage conditions of seeds and fruits, but also the processing time have a major impact on the quality of the oil. Bad storage conditions, for example, can lead to the presence of mycotoxins. Some inappropriate drying conditions can lead to the formation of polycyclic aromatic hydrocarbons like benzoapyrene. And afterwards, the way seeds and fruits are treated for oil recovery is also of prior importance. They can be pressed or solvent extracted, and if too high pressure or temperatures are applied during the extraction, this can lead to the leaching of some undesirable components, like polar oxidation compounds, for example. And then, once the oil is obtained, the storage time conditions play a major role on the quality of the final product. And finally, purification process and the parameters, if not properly conducted, can also lead to the formation of undesirable compounds, like trans fatty acids or glycidyl and 3-MCPD esters. As an illustration of the geographic variability, you can see on this graph the variation in the alpha-linolenic acid content, which is of soybean oil. The alpha-linolenic acid is one of the fatty acids contained in soybean oil. And you can see the variation of this content all over the years for the crude soybean oil. From October to April, the soybean oil is sourced from crops grown in the North Hemisphere, so it's represented in light blue. And oil coming from this area has a higher content in alpha-linolenic acid, with an average around 7.5, compared to soybean oil sourced from crops grown in the South Hemisphere, for which the average content is 1 point less, around 6.5. And you can see that even for each geographic origin, we can have a batch-to-batch variability that can be sometimes high. Another example with olive oil. The fatty acid composition is also very dependent on the cultivar. You have here represented a graph showing the average content of the oleic acid, which is the major fatty acid in olive oil, for 4 different cultivars, all grown in the same region in Tunisia. So you can see that the oleic acid content can vary from 60 to 74%. That's why the standards on extra virgin olive oils that are edited by the Codex Alimentarius are so broad, with specifications ranging from 55 to 83%. So now let's have a look at the different existing standards we deal with. On one hand, we have the entire and wide range of what the nature can provide. These available food-grade products are supported by the Codex Alimentarius guidelines. And on the other hand, we have to comply with pharma regulatory standards, like the various national pharmacopoeias, often tighter than food standards. And finally, with even narrower and specific customer requirements that may have very particular needs, even on parameters that are not mentioned in the pharmacopoeia monographs. So here is an example on olive oil of what can be the differences between food standards, pharmacopoeias, and the customer's requirements. For oleic acid, we saw that the food standard is between 55 and 83%, whereas the specifications for the pharmacopoeia are between 56 and 84% and 85%, sorry. And above that, one customer will need a product complying with USP. Meanwhile, another one will need a product with tighter specification, as in the case of customer 2. And things are becoming even more tricky when these specific requirements are combined on several fatty acids. You have here the example of customer 2 that combines specific requirements both on oleic and linoleic acid. Now let's have a look at how we manage that variability from the raw material to the finished product to guarantee the consistency of our pharmaceutical ingredients. So first, and of a major important step in this management is the sourcing of the starting material. Not all vegetable oils are the same, as we saw previously, the importance of many parameters on the variability of the product. And for us, it's really essential to work in a very close collaboration with our suppliers to discuss, to explain clearly what are our needs, to be sure that pharma requirements are well understood, to ensure quality and consistency of the starting material. So we select our suppliers regarding our very specific requirements, like the fatty acid composition, for example. So it can be suppliers working with a specific crop cultivar from a specific geographic area of cultivation, or a particular supply period over the year to guarantee the freshness of the oil. It can also be a supplier working with dedicated lines to avoid cross-contamination. And we also work in close relationship with our suppliers to help them improve their process, to avoid oil damage and guarantee the high-level quality of our pharmaceutical ingredients. Prior to the supplier audit and the approval, we have a phase of pre-approval where samples are deeply characterized by our QC and R&D lab. And once the supplier is approved, it is regularly audited. And for each production batch, we receive a pre-delivery sample that is characterized by the QC lab to have the agreement prior to the delivery of the starting material. Now, let's have a look at the composition of a vegetable oil that is used as the starting material. So the starting material can be crude or virgin vegetable oil. We talk about crude oil when the oil is extracted with a solvent. That's the case, for example, for soybean oil. And we talk about virgin oil when the oil is obtained only by pressure. It's the case for olive oil. Before the purification process, crude or virgin oil contains a lot of different compounds. Triglycerides are the major components, and they represent the large majority of the vegetable oil composition, with more than 95%. And the oil can also contain little amounts of diglycerides and monoglycerides. Among this, we have some minor components representing not more than 5%. And we can find in this category some desirable components like tocopherols or polyphenols that are the natural antioxidants from the oil, and they protect the oil against oxidation. And we can also have some undesirable ones like the pigments, flavors, phospholipids, free fatty acids. And apart from these components, we can have some contaminants, two types of contaminants. Some coming from the starting material that are mainly from culture origin, that can be pesticides, dioxins, PAH. And we have also some contaminants that can be generated by the process if it is not well conducted. That's the case of oxidation compounds, trans fatty acids, some fatty acid polymers, and esters of 3-MCPD and glycidol. So it's important to notice that these minor components are not to be considered as contaminants, as they are part of the typical composition of the oil. And vegetable oils need to be purified to comply with pharmaceutical quality standards. And the role of the purification process is to remove contaminants and undesirable substances, avoid the formation of process contaminants, while preserving natural antioxidants to guarantee the perfect safety, quality, and stability of the final product. So now let's go a little bit deeper in the purification process of the vegetable oils. The major step of this process are degumming, neutralization, bleaching, and deodorization. Degumming step first, it's a step that is needed for oils that are rich in phospholipids. It's not the case of all the oils, but it's the case, for example, of soybean oil, not for olive. But the aim of this step is to remove the phospholipids. And in addition, it also allows to eliminate some trace metals, hydrophilic contaminants, and polar oxidation compounds. Once the oil is degummed, it can be neutralized. So this neutralization step aims at removing free fatty acids and traces of phospholipid that are left from the degumming step. But this step also allows to decompose some pigments, like chlorophyll, that will be then more easily removed during the bleaching step. As neutralization, bleaching step is a critical step in the process to ensure quality and stability of the final product. It aims at removing natural pigments that are responsible for oil coloration, but also trace metals that can act as oxidation catalysts. This step can be done with bleaching clays containing or not a part of activated carbon. And these processing aids are used in variable quantities depending on the nature and depending on the nature of the oil and the compounds to be removed. We also adapt at this step contact time and temperature to adjust depending on the type and composition of the oil we have to treat. Last step of the purification process is the deodorization. It allows to remove remaining free fatty acids and oxidation compounds that are responsible for taste and odor and can be damaging for the good conservation and stability of the product. This step allows to reach very low levels of acid and peroxide values, so to guarantee the good stability of the final product. So a large part of the oils, vegetable oils that are used today as excipients for drug formulation are generally food-grade oils. Just here to show you some major differences between our process and the typical refining process that is applied to produce edible oils. First, most of the edible oils are refined using a continuous process and can be either chemically or physically refined. We apply a closed batch process purification under high vacuum and nitrogen to avoid any contamination and oxidation of the oil. We also use special quality of bleaching clays and activated carbon, and in much higher amounts compared to what is done in the food industry. And finally, regarding the deodorization step, we work at much lower temperatures to avoid any degradation of the oil and keep as possible the natural antioxidants in the vegetable oils. As an illustration, you can see here that our process is highly respectful of the product and allows to avoid the degradation of the oil and especially the formation of trans fatty acids, while keeping a high amount of tocopherols that are the antioxidants that are naturally present in the vegetable oil. In addition to the sourcing of the starting material, the process management is a key point to manage the variability of plant-based pharmaceutical ingredients. Analysis of the starting material composition, adjustment of the purification process parameters, and real-time process monitoring allow us to fine-tune our process conditions to guarantee the best batch-to-batch consistency. And in process analytical control tests, ensure that every batch is compliant with our internal specifications, and how out-of-trends monitoring enables us to identify immediately any process deviation. We aim to be as consistent as possible in the production of our pharmaceutical ingredients. Here is an example of the batch-to-batch consistency on the acid value, which is a critical quality attribute for injectable grade soybean oil. So we can see that the values on the finished product are very narrowly dispersed, especially if we compare to the values on the starting material in blue, indicating an efficient and well-controlled purification process. And the accepted limit on the final product is here shown in red. The stability of our pharmaceutical ingredients is also tested according to the ICH Q1A guidelines. in storage conditions that are representative of the different climatic conditions of the countries where we ship our products. The graphs here show the results of stability studies on 3 validation batches regarding the main critical quality attributes that are acid value and peroxide value. And we can see that they are perfectly stable over a 36 months shelf life period. And by way of comparison, the shelf life of food grade oils is generally of 1 year. So as an API manufacturer, we have a strong pharma culture, and we are well aware of the specific requirements of pharma industry. Our active ingredients and excipients are produced by trained and experienced employees. We operate in line with the required GMP standards, HIPEC for excipients and ICH-Q7 for APIs. Our production site is regularly inspected and audited by the relevant health authorities like ANSM and US FDA. And we have been granted the GMP certificates and attestations for our APIs and excipients. And each day we commit in bringing the same care to excipients production as to APIs. Our products are regulatory supported. We have submitted regulatory files to the major international health authorities for our pharmaceutical ingredients. So we are, for example, the only company in Europe having a certificate of suitability for injectable grade olive oil. And we also have one for injectable grade soybean oil. For the US, we have submitted DMF Type 2 for refined soybean oil and olive oils for a dedicated customer. But we have also DMF Type 4 for soybean oil and sesame oil. And a DMF Type 4 for olive oil will be submitted to the FDA by the end of the year. We are also registered in India and are submitting DMF for olive and soybean oil also in China. It's in progress. So, as a conclusion, we saw the importance of managing the pharmaceutical ingredients variability to ensure drug therapeutic effectiveness, and natural-based products are by definition subject to a certain degree of variability, and this variability can have many different origins. So making pharmaceutical ingredients from these natural-based raw materials can be sometimes very challenging. But it's made possible by a rigorous sourcing of the starting material and perfect control of purification processes and the GMP requirements. And we can provide you a wide range of consistent vegetable oils for use as APIs or as excipients.